Hello, my name's Toren Finkel, and I'm one of the co-founders and the Chief Scientific Officer of Generian. Generian is a Pittsburgh-based startup that was capitalized 6 months ago with a $20 million Series A investment from UPMC Enterprise. Enterprise is the $1 billion venture arm of UPMC, which is a payer-provider system here in Western Pennsylvania. Today, I'd like to give you a brief introduction to the science behind Generian, the considerable progress we've made to date, and the exciting near-term goals I believe we can achieve. The central organizing principle behind Generian is to use small molecules in order to amplify natural signaling networks in order to reverse the course of disease. To give you a little better context of what type of signaling networks we're targeting, it's important to know that the scientific underpinnings of Generian comes from the lab of the three founders, all of whom are members of the Aging Institute at the University of Pittsburgh and UPMC. As many of you probably know, aging is the predominant risk factor for most diseases. And over time, our cells no longer function as they should, and that in turn alters signaling networks that regulate properties like nutrient sensing or intracellular quality control. Work from our labs, and as well as many other labs, have defined evolutionarily conserved pathways that regulate this type of signaling networks, and which in turn have been shown to regulate the overall lifespan and health span of organisms as simple as the worm C. elegans to human beings. And in essence, Generian is leveraging the insights from aging biologies to develop therapies that amplify these naturally signal networks in order to reverse the course of disease. The two proteins that are now part of Generian are shown here. The first is the protein AMP kinase, which is an intracellular kinase involved in nutrient sensing and cell metabolism. It's a key regulator of several critical metabolic pathways, making it a potential therapeutic target for treating multiple diseases characterized by impaired metabolism. A second target is TFEB, which is a transcription factor involved in cell intracellular quality control through its regulation of processes such as autophagy and lysosomal function. TFEB is a transcription factor that orchestrates the expression of 400 downstream genes. It has been implicated in various diseases known as proteopathies, which are caused by what happens when proteins are structurally abnormal and form toxic aggregates that in turn disrupt the function and viability of cell. As such, TFEB is a promising therapeutic target for a number of diseases caused by these aggregates, be they proteins or lipids, or for diseases in which there is impaired lysosomal function. Although the work from our lab told us which proteins and networks to target, we did face a number of challenges. First, our observations and those of others suggested that as we age or suffered from disease, the activity of AMP kinase and TFEB declined. And so what we needed were small molecules that activated these pathways. That's unusual for small molecules because generally drugs work by inhibiting their targets, not by activating them. A second challenge is that although we wanted to turn on these pathways, we didn't want to do that in all tissues at all times, but only turn them on where and when they were needed. In essence, we wanted to preserve and not override the natural physiological cues that regulate these networks. The solution to these challenges came from the realization that E3 ubiquitin ligases are powerful tools to modulate protein activity. Some of you may be aware that E3s are currently being used in a strategy called PROTAC in order to degrade harmful proteins. But we at Generian believe they can also be used to increase the activity of beneficial proteins, thereby restoring the activity of these important signaling networks and improving overall cellular function. In essence, Generian is using inhibitors of specific E3s to prevent the degradation of beneficial target proteins in order to do our core mission, which is to help people live better, longer. The basis for this strategy is shown in a little bit more detail here. Work from our lab has demonstrated that for both TFEB and AMP kinase, once these proteins are activated by their normal physiological cues, they are quickly degraded by specific E3s. We have identified these E3s and developed small molecules that block their function. By doing so, we maintain the physiological activation of both TFEB and AMP kinase. Again, this shares some conceptual overlap with PROTAC, which is using E3s again to degrade harmful proteins, while we're engaging E3 ubiquitin biology to amplify and maintain the levels of helpful therapeutic proteins. How do we do this? In general, we achieve this through our small molecule Network Activator Platform, or SNAP. This platform enables the rapid and scalable target discovery through the use of proprietary tools and assays that pinpoint the relevant target protein, identify the specific E3 ligase, and develop small molecules for future studies. This in general employs a systematic and unbiased approach to identify the E3 using a high throughput validation process. Once the E3 is identified, we use a secondary proprietary screening assay that allows us to analyze millions of small molecules for their ability to block the specific interaction between the E3 identified and the target. To give you some example of how this might work in vivo, here's an example from early on from one of the tool compounds in our TFEB program, in which we've given normal rats 4 doses of the TFEB activator, either a vehicle or the TFEB activator, and look, in this case, for evidence of increased lysosomal number by looking at the lysosomal marker cathepsin D, or LAMP1. We're looking, in this case, in a specific region of the brain where tyrosine hydroxylase, or dopaminergic neurons, are seen. As you can see here, a short course of TFEB activator results in a marked increase in lysosomal number in these animals. Not only do we see increase in number, but in subsequent experiments, we could show that there is more lysosomal activity, and in fact that this led to increased resolution and removal of harmful proteins like alpha-synuclein. As at this time, Generian is composed of 2 programs, as I mentioned, TFEB and AMP kinase. Both of these programs had an extensive MedChem campaign that involves the synthesis and testing of over 300 novel structures, compounds, and both programs we anticipate to be in phase 1 studies by 2021. I should mention that both AMP kinase and TFEB are viewed as difficult drug targets, and in particular, transcription factors like TFEB are in general viewed as undruggable. However, we're very convinced that our SNAP platform provides a way of drugging transcription factors, and as such, we have a number of ongoing programs exploring the possibility of drugging other transcription factors that might have therapeutic benefit. What are our clinical indications? As I mentioned, for TFEB, we're very intrigued by the observation in a number of preclinical models that TFEB activation leads to the clearance of protein aggregates. And so we're evaluating both aggregates in the CNS, for instance, diseases like Parkinson's or Lewy body dimensions, or rare conditions in the periphery that might be more approachable, such as certain skeletal myopathies, which are again caused by toxic aggregates. We do have other programs, preclinical evaluations going on in rare indications like lysosomal storage disease, and in more common but unmet needs like dry AMD, all of these conditions in which autophagy or lysosomal dysfunction have been implicated. For AMP kinase, our lead indication is polycystic kidney disease, where again there's a wealth of preclinical data from other laboratories suggesting that activation of AMP kinase provides a strong inhibitor of cyst growth and expansion. However, like for the case of TFEB, we are evaluating other models at this stage, both common conditions like diabetic nephropathy and rare conditions like myotonic dystrophy. The success of any company rests not only on the underlying science, but also the execution of the team. We're fortunate to really have a great team at Generian. I mentioned that I'm the Chief Scientific Officer and one of the co-founders. I'm joined in the company by my other 2 co-founders, Yuan Liu and Bill Chen, who bring decades of experience in the biochemistry and chemical biology of E3 ubiquitin ligases. We're led by our Hank Saferstein, our CEO, who has a proven track record in leading organizations, M&A, and fundraising. He's raised over $250 million in the past from both private and public equity markets, taken 2 companies to IPOs and 1 company to a corporate acquisition. Although we're based in Pittsburgh, we have a Boston footprint which is headed by Valerie Cullen, who's our VP of Translation and Development. In addition to the in-house team, we have a great SAB, including Anna Maria Cuervo, Jerry Schulman, and Ruben Shaw, who are experts in autophagy and AMP kinase and metabolism biology. These are very distinguished scientists, many in the National Academy, and all of who provide incredible advice as we go forward. So in summary, Generian, I think, provides a unique investment opportunity. We're developing first-in-class small molecules that can augment the activity of TFEB and AMP kinase. Both of our programs are in the IND enabling stage, and with the hope and expectation that they would both be in the clinic by 2021. I didn't have time, but both programs are backed by a rigorous blood-based biomarker program that will allow us to assess target engagement in healthy volunteers. I mentioned at the onset, we've raised $20 million already, and our expectation is to extend the Series A to achieve our goal of $55 million. We have also had preliminary discussions with pharma, and that might be a possibility of early partnering with one or of these pharma partners for one or both programs. So I hope that gives you a little flavor of what we're doing at Generian. I hope it's of interest. If it is, please feel free to contact our Director of Operations, Molly Curran, and I do look forward to talking to you in the near future.